Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about a very special group of bacteria which is known as Mycoplasma or molecules. The whole group is called as molecules. Molecules actually a group of eubacteria or true bacteria. The common name for this group has traditionally been Mycoplasma. So earlier the name Mycoplasma is used to designate this group. However, this usage invites confusion. The use of Mycoplasma for this whole group creates confusion, since it is often not clear whether Mycoplasma is representing a genus or the whole group. To avoid this confusion, the more recently coined term molecules to designate the members of eubacterial group is being adopted here. So to avoid this confusion whether Mycoplasma is a group or a genus of the whole group, the scientists term or coined a term molecules to designate a very special group of eubacteria. The distinguishing property of the molecules is lack of a definite cell wall in them. They all are sensitive to osmotic lysis as they lack a tough or hard cell wall, and they are resistant to penicillin. That means penicillin cannot kill or destroy the cell of these molecules. Then other wall synthesis antibiotics also cannot have any effect on Mycoplasma or molecules. Pleomorphic in same shape to at least some extent. So these are pleomorphic. Pleomorphic means they can change their shape according to the situation or according to the environment. And these are parasites to eukaryotic organisms. That means they live as a guest on a host inside the host body. Then their colonies that develop on solid culture media are often small, typically having a nipped or fried egg appearance. So the colony of Mycoplasma appeared as a fried egg. The molecules have unusually low guanine and cytosine value, and they have very small genome. 16S ribosomal or rRNA sequencing data reveals that molecules are coherent polygenic group closely related to Clostridia, and this molecule group have 6 genera. One is Mycoplasma, second Phytoplasma, then Ureaplasma, then Acoleplasma, then Anaeroplasma, then Spiroplasma. So these are different groups of molecules, and this table represents different characteristic features of the genera which are included under the group molecules. Like morphology, Mycoplasma are pleomorphic in nature, but Ureaplasma is cocci shaped, that means round shaped. Again, Archoplasma is pleomorphic, that means their shape changes according to the situation or environment. Anaeroplasma is cocci shaped, Spiroplasma is helical shaped. Okay, then habitat, motility. So different genera have their own characteristic features. Okay. So here we will discuss only about the Mycoplasma. These are typical prokaryotes, being smallest known organisms which are capable of autonomous growth and reproduction. The smallest viable mycoplasmal cell known is Mycoplasma hominis S39 or S39, which is about very small 0.33 micrometer. This is only about 2 times larger than theoretically considered smallest cell containing all informations. A highly contagious disease of cattle appeared in Germany and Switzerland in 1713. In the year 1713, and it spread throughout the throughout Europe during 18th century. This disease was named as bovine pleuropneumonia, and its causative organism was thought to be pleuropneumonia like organism, PPLO. And this discovery was made by Knockard and Rocks in 1898, and the organism discovered was given the first binomial name as Asterococcus mycoides. Later in the year 1929, Novak replaced the generic name or the name of the genus by Mycoplasma. So later on, Novak in 1929 replaced the genus name Asterococcus by Mycoplasma, and latter is now generally used in the place of PPLO or pleuropneumonia like organism to represent these organisms. Okay, so it is all about the discovery of Mycoplasma. Next, so this is the cell structure. This is the structure or cell structure of mycoplasmal cell with plasma membrane, cytoplasm. Then these have ribosome, then soluble RNA, soluble protein, DNA molecules, and they lack cell wall. Okay. Mycoplasma are non-motile and do not produce any spores. They are important pathogens of animals though they may occur as saprophytes. So generally they are pathogens, but sometimes they occur as saprophytes. That means they grow on the dead and decaying matter. At present, more than 36 mycoplasmal representatives have been isolated, and most typical representative of the pathogenic species are causative agent of pleuropneumonia in cattle, that is Mycoplasma mycoides, and acute respiratory infection which is caused by Mycoplasma hominis, and a typical pneumonia in human which is caused by Mycoplasma pneumoniae. So these are causative agents of different diseases. Mycoplasmas lack cell wall and are highly pleomorphic. They can change their shape from spherical to branched to filamentous and multiply generally by binary fission. What is binary fission? One mycoplasmal cell which is regarded as parent cell will divide it to form two daughter cells, and it will not follow any rule, and they divide by amitosis. Okay, and the whole process is known as binary fission, where one cell will divide to form two cells, two daughter cells. The cell organelles which is present inside the cell of the mycoplasma, at first the plasma membrane, then ribosomes, DNA and RNA. The plasma membrane which binds mycoplasmal cell in absence of cell wall is tri-layered and contains mostly cholesterol. The genome or DNA of the known mycoplasma are large enough to code only for about 600 to 1000 polypeptides. It is this limited information which limits the ultimate complexity of mycoplasma. So the DNA or genome of mycoplasma contains very limited information. They can code only 600 to 1000 polypeptides. So the latter are resistant to wall attacking antibiotics such as penicillin, because as we all know that mycoplasma lacks cell wall, so they are resistant to penicillin, because penicillin actually destroys the cell wall of bacteria. So as mycoplasma lacks cell wall, so the penicillin cannot have any effect on the mycoplasma. The growth of mycoplasmas is inhibited by tetracyclines and similar antibiotics which act on the metabolic pathways instead of cell wall. So this is all about mycoplasma, and I have discussed all this from a book, from a microbiology book by R.P. Singh, and I hope you really enjoyed this session. If you liked it, please hit the like button, share with your other friends, and subscribe to my channel Sunanda's Tutorial. Thank you. So before ending my lecture video, here I have mentioned some diseases caused by the mycoplasma, the pathogenicity of mycoplasma for animals. So mycoplasma mycoids causes pleuropneumonia in cattle. Some strains of this mycoplasma produce pleuropneumonia in goat. Mycoplasma bovigenitalium causes inflammation in genitals in animal. Another species, Mycoplasma ajalactia, produces ajalactia of sheep and goat. Then mycoplasmal disease of plant. In plants, mycoplasma produce disease usually by upsetting the hormonal balance. This imbalance is expressed in form of symptoms such as dwarfness, loss of apical dominance, enlargement of floral buds, and bunchy growth of newly formed sprouts. In plants, the mycoplasmal disease are transmitted by insect vectors belonging to the class of Psychedeliaceae and Phyllidae. Multiplication of mycoplasma within insect body has been demonstrated in many experiments. Insects fed on mycoplasma infected plants become infectious only after a specific latent period. Some common mycoplasmal diseases of plants are little leaf disease of brinjal, clover dwarf, clover phylloide, clover virescens, cotton virescens, bunchy top of papaya, witch's broom of legumes, stripe disease of sugarcane, big bud of tomato, and yellow dwarf of tobacco. So please remember these diseases. It is very important for your examination.